Today it's really a privilege to introduce Roger. Really for this group probably, well, there's a lot of young people here, so they do need an introduction, but Roger is one of us. I didn't realize until looking at his CV, but Roger did his undergraduate work in physics. And then he went on and got his Ph.D. in physiology from the Medical College of Virginia, and then did a postdoc at University of Western Ontario. And he came to San Antonio in '72, and he came here before there was a physiology department. And I've heard that the reason they built the physiology department was because of Roger. And then Roger then recruited Ed Masro to be chair of this department that he had created. And actually, I don't think Roger was involved in aging before Ed came, right? And he became involved with aging and was part of the nutritional probe on aging process, the dietary restriction program project for, I think it was about 20 years that it was funded. And he'll be talking about some of this research because one of the important observations that Roger made, which he had to constantly argue for, was the effect of dietary restriction on metabolism, and I'm sure he's going to be talking about that today. But I just wanted to emphasize is that not only is Roger a great researcher, but he's an outstanding teacher, you know, and also he's a politician. I was just looking through this. He's has been past president of the American Aging Association, past president of the Gerontological Society, past president of the Pan American Congress of Aging, and I'm surprised he isn't running as a candidate for the Republican president here. And in 2009, he received the Presidential Medal for contributions to the development of gerontology by the International Association of Gerontology and Geriatrics. So it's really great to have Roger. Oh, I forgot to tell you, Roger left us about 8 years ago for Penn State. And so this is the first time I think we've had you back for a seminar. So it's really great to have Roger back.
Thank you very much, Arlan. I particularly like the rewrite of history in terms of the development of the Department of Physiology, because I can tell you that when Ed Masro first took the job in '73, and I'd been a faculty member for 1 year, he took a look at what I was doing, which I dearly loved. I mean, I was a muscle physiologist, and I just loved it. And he said, McCarter, if you want to survive in this department, you better start doing aging research. I got the message, I got into aging research, and I learned to love that too. So I'm nothing if not flexible. Anyway, what I'd like... The truth always hurts. So what I'd like to do today is to share with you some of the reflections I've had in just the past couple of months. And it was heavily influenced by the popular book by Rebecca Skloot called The Immortal Life of Henrietta Lacks. This is the story of the woman who developed cancer, and her cells were used without her permission to develop the HeLa cell line, which has been used ever since to great benefit for many, many people. And it made me think back to the years of going to aging meetings and seeing the battles raging between those scientists like Jim and Olivia Smith who were studying aging in cells in vitro versus the rest of us who are doing aging in vivo, looking at the intact organisms. And at one particular conference, which was attended by a very large number of people, and most of the experts in the area were there, the consensus of opinion was that actually you were never going to learn anything much about real aging by studying aging of cells in dishes. Well, I think we've all come to appreciate that a great deal has been learned about aging from studies in vitro, as well as in vivo, of course. So both are true. And I think that is true of so many of the theories of aging. When I look back on the past 40 years of being involved in this activity, it's really quite something to remember the so many theories which have come and gone, been discarded, and then come back into prominence as we start learning more and become more educated. And I was particularly reminded of this at the recent GSA, Gerontological Society of America conference in Boston just a couple of months ago, when Steve Ostad, in his award-winning talk, mentioned that the rate of living theory of aging was impossible to kill. That this is a theory that kept coming back no matter how good the evidence against it was. And there seem to be very many theories in this category. And at the very same meeting, a very distinguished metabolic physiologist from the Pennington, Eric Ravusin, presented his data on men and women on calorie restriction, which he proudly pointed out was consistent with the rate of living theory of aging and supported that theory. So this idea is one of those which are so popular that they will be with us for a very long time. Okay, so moving right along, I did want to recognize the very large number of truly terrific people. It's been a joy working with all of these people over the years. And I've learned so much from all of them, those who've been directly involved in a lot of these studies. And there we go. I'd also like to acknowledge these investigators who've very generously shared their slides with me and permitted me to use them in this talk. Now, we all know the phenotypic expression of aging. And the big question which we're all trying to address is what are the mechanisms that produce the degenerative changes of aging? One of the most popular mechanisms over the last 5 or 10 years has been Sirtuin genes associated with resveratrol and the whole red wine business. Can we drink a whole lot more wine and be healthier and happier and live longer? And this article appeared in Science a couple of, I think it was a month ago, the Sirtuin story Unravels. So here's yet another popular theory of aging, which is coming under attack. And one of the proponents who originally discovered the theory or discovered these genes, Matt Kaberlein at Washington, made the comment that this mammalian Sirtuin hype, thank you, is based on worm and fly work, and much of that work is now looking questionable, to which David Sinclair, who sold his company to GlaxoSmithKline for $700 million, made the comment that rumors of the death of Sirtuins and aging are greatly exaggerated. And Felipe Sierra, trying to play the diplomat in this whole crowd, made the comment that all of these very different results are likely to be correct. So it made me think that what if Felipe Sierra, despite trying to be diplomatic, in fact was correct? Is there a possibility that, in fact, all of those results may indeed be accurate? Fantastic, thank you. So, one of the theories I immediately thought of is one which had really fallen out of favor, in fact, not even been discussed for many, many years, and that was the theory, the immune theory of aging, proposed by Roy Walford in '62 and '69, which came under, made me really sensitive to Rita Efros's wonderful talk at the GSA, where she pointed out that senescent T cells were really the canary in the coal mine of age-related degeneration. Because these senescent T cells, not only were they no longer beneficial in terms of killing off antigens that invaded our body, but they sat in our tissues and secreted inflammatory cytokines which were harmful to us. And this point was driven home by the manuscript of Cheryl Conover, who had genetically engineered mice so that they would not undergo thymic involution, and so they would no longer have, age-related mice would no longer have large numbers of senescent T cells. And the prevention of thymic involution greatly slowed aging in these mice and increased their longevity. So they were healthier and lived longer. And finally, this was driven home by Judy Campisi in 2011, who pointed out that most senescent cells, in particular in skeletal muscle, adipose tissue, and kidney, developed what she called a senescence-associated secretory phenotype, or SASP, and that this was involved in aging of tissues because, again, these senescent cells were hanging around not just not doing the things that they had originally been intended to do, but were now actively working against the maintenance of homeostasis. In other words, they were deleterious. And the recent paper by Van Dersen et al., which appeared in Nature but was featured in the New York Times, involved the purging of senescent cells from muscle and fat and kidney, and the animals in which the senescent cells were removed were remarkably healthier and lived longer, admittedly in a cell in a mouse line which had short longevity, but nevertheless a dramatic confirmation of the sort of thing that Judy Campisi had been talking about. So here is a theory whose time had come and gone, and then suddenly, and these are the mice from the Van Dersen et al. Study. Now, of course, we're all very familiar with one of the great theories of the last 40 years on why we get old, or is it correct or not? The free radical oxidative stress mitochondrial theory of aging, Denham-Harman in 56-72, and then certainly pushed by Raj Sohal and Gustavo Barha in 93 in the early 90s. Oh boy. Predictions, we're not going to be able to see the predictions. This is one of Holly's slide, which is actually a great slide if we'd been able to see it. But the predictions of the oxidative stress theory of aging, oh, here they come. And I'd like to focus on prediction number 3, manipulations that alter oxidative damage and oxidative stress will alter life. And of course, by just terrific studies done here in San Antonio at the Barshop, it was shown that over or underexpression of antioxidant enzymes had no effect on longevity at all. And they pointed out that these results really undermined the belief in the oxidative stress theory of aging. However, that oxidative stress certainly may play a role in animals in a stressed environment. So another theory that I try to address as well in my research was the glycation theory of aging. Anthony Cerami, 1985, had suggested that diabetes could be a model of accelerated aging, that diabetics in general live a lot shorter than people who are non-diabetic, and that this may be associated with the higher levels of glycemia, with the rationale that plasma glucose leads to enhanced glycation of proteins, promoting oxidative stress, and both of these increase pathology, decrease function, and so promote aging. And our studies in transgenic mice, which overexpressed the glucose transporter, GLUT4, showing that normal control mice, as well as the GLUT4 mice fed ad libitum, ate the same amount of food and had the same characteristics of body weight, and the transgenic animals that were fed the restricted diet had exactly the same trajectory of body weight with age as control animals, and all of these mice lived the same. So apparently, even though glycation reactions probably play a major role in the pathologies associated with diabetes, the high levels of glycemia were certainly not one of the mechanisms of aging associated with calorie restriction. Now, one I'd like to spend some time on is the granddaddy of them all, the rate of living theory of aging. This is a particularly interesting one because it was started by the data generated by Max Rubner in the late 1880s, so a long time ago. Max Rubner was a very good scientist, and he measured the oxygen consumption of a large number of domestic species, and here we see horse, cow, dog, cat, pig, and he found that the oxygen consumption per gram of body weight over their lifespan was approximately the same over their whole lifespan, with men and women being an exception, having a high. Nobody bothered about the exceptional properties of men and women because they believed that men and women were exceptional anyway. So they focused on the similarity of these data, and it led to the idea that most living things have a certain amount of calories that they can process in their lifetime. And George Sacher seized on this idea by saying that the mechanism of calorie restriction in rats in prolonging life is a consequence of the decrease in the rate of energy metabolism. And this was a wildly popular theory, and it had a lot of experimental basis too, because it had been known since the 1920s that basal metabolic rate decreases when you eat less to a greater extent than loss of body weight can explain. And an English endocrinologist pointed out in 1978 that this effect is pervasive. There is no investigator who has looked for this effect and failed to find it. So the theory, rate of living theory of aging, and the explanation of calorie restriction had a great deal of experimental support. Now, Ed Masra and BP and Helen Bertrand published some results in 1980 showing that the food consumption per gram of body weight was similar in calorie-restricted animals and animals fed ad libitum. And that didn't seem to bother people very much. I don't think you caught a lot of flack with that, Ed. But certainly when we came out with our oxygen consumption data showing that the oxygen consumption of animals fed a restricted diet, male Fisher 344 rats, was certainly lower than animals fed ad libitum, but if you kept them on calorie restriction for longer than 8 weeks, then the metabolic rate per unit body weight normalized so that all of the animals had essentially the same caloric output per gram of body weight over their lifespan. Now, this was extremely unpopular because people knew that the rate of living theory was true, and it did explain how calorie restriction worked. So I can vividly remember going to meetings and being denounced by people who simply said they didn't believe the data, Rick Weindrup being one of them. To my consternation, announced at a national meeting that he simply did not believe the data. So, being totally perturbed about this, we repeated the experiment. And we did this now, allowing the animals, one group of 4 groups of animals, to have access to exercise wheels so that they could run whenever they wanted to. And the results were spectacular. The effect of exercise in the case of the restricted animals was dramatic. The animals fed 40% less were running up to 3.5 miles a day at a time when all of their genetically similar brothers, they were all males, were dead. So here are animals behaving like I do, eating ad libitum, exercising very little, and dying at before your prime. But here are the restricted animals, and they did very well, and here are animals on a 10% restriction. So the effect of restriction was to increase physical activity, and this increased physical activity in the face of a limited caloric input meant that the body weight dropped dramatically. So the caloric output per unit of body weight, which we were measuring simultaneously, was very, very high. And to the consternation of the rate of living theory people, the animals who had a very high rate of metabolism actually lived, if anything, a little bit longer than those animals which were sedentary and not allowed to exercise. And here are the ad lib with low levels of physical activity dying at exactly the same rate as those animals which were sedentary. So the results were very unpopular, and it became more so when a number of different investigators found that with the animals that they were using, the rate of metabolism actually decreased in caloric restriction and stayed down. In other words, there was a sustained reduction of metabolic rate over the lifetime of these animals. On the other hand, John Speakman and his collaborators published results showing that in the particular animals that they were studying, those animals fed less, fed the calorically restricted diet, actually had a higher rate of metabolism per unit body weight than ad libitum fed animals. So 3 totally different results in response to caloric restriction. Now, David Sinclair, in a review article, pointed out that this field is in a real mess. But my response to that was that the field was only in a mess if your prejudgment was that the rate of living theory had to be correct. In other words, what if all 3 results were actually correct? That the response to calorie restriction could induce either an increased rate of metabolism per unit body weight, a decrease, or no change over the lifespan. And so that seemed to be a cop-out to most people, but nevertheless, that was the stage that was left at. And I believe, actually, fundamentally, that all of those results are indeed correct, and that calorie restriction, as one of its characteristic features, has a metabolic response which can either be an increased rate of metabolism, a decreased rate of metabolism, or no change over the lifespan. In other words, yeah.
Could you just give a couple of examples of where you see these 3 different responses?
Microphone?
There we go. Could you give us a couple of examples of where you might see these different responses to caloric restriction?
Yes. So, for example, our studies were done in male Fisher 344 animals, obviously kept behind a barrier. There was a study by, I think Sohal recently published a study in which he measured metabolic rate intermittently, but it remained low. I think that was in C57 black 6s. And then Speakman's crowd, as my recollection, were using voles. And if I'm correct on that, Steve, I think they were using voles. And in other words, there were 3 different species involved. And I think this is the key factor, which I'll come back to later on, that these were 3 different animals, 3 different genetic backgrounds. And so one of the messages I'd like to get across is that we need to be very, very sensitive to the effects of genetic background in looking for the particular phenotypes, and that we, as grown-ups, cannot expect the same phenotypic response to an alteration in the environment all the time, that it's going to be different depending on the circumstances. So anyway, I thought that Steve Ostad brought a great deal of clarity to this discussion by his general observation that organisms, because of anatomy, physiology, or environmental conditions, are sheltered from extrinsic hazards, will evolve slow aging. So these different types, like Shelley's naked mole rats, I guess this is a porcupine, Steve, and of course we have a bat here. So all of these different species have evolved different mechanisms of extended longevity as a consequence of environmental conditions. And I was particularly taken by the study of Jobson et al., Molecular Biology of Evolution, in 2010, who scanned 25 mammalian species and found that genetic targets of natural selection for elongated lifespan in mammals are cellular membrane and extracellular collagen composition, not genome integrity. So the integrity of the genome is not something that is focused on by natural selection in evolving extended longevity, that there are other factors which are more important. And this is borne out in the study that my colleague at Penn State did in Swedish twins, Jerry McLaren and others, looked at Swedish twins and similar results by those who were studying Danish twins, and these were identical twins, monozygotic twins. And their conclusion was that the data indicate that the heritability of longevity over the entire age range is low. A maximum of around 1/3 of the variance in longevity is attributable to genetic factors, and almost all of the remaining variance is due to individually unique environmental factors. So that the individually unique environmental factors play a major role in longevity and, at least for my money, therefore health over the age span. So where are we? I think we're certainly not at the beginning, because a great deal of terrific work was done. And in particular, I want to credit Ed Masro, who started the aging program here at the UT Health Science Center in 1973, for being a catalyst for the development of what has been the spectacular success of the Barshop Center under Arlan's direction over the last several years. And I think we need to be very mindful of Albert Szent-Györgyi, Nobel Prize winner for the discovery of vitamin C, for his observation that for every complex problem, there is a simple, easy to understand, and incorrect answer. Part of the problem is we like to look for simple solutions. And very often, when we're looking at complex phenotypes, it is simply not reasonable to expect a simple answer. Longevity and aging are simply complex phenotypes, and we should not expect simple answers. So, our gold standard for the past 50 years has been the discovery by Clive McKay in 1935 of the fact that animals fed less food over their lifespan live a great deal longer. And I think we can all agree on the fact that calorie-restricted or dietarily restricted animals are in a different metabolic state with all of these interesting properties in one strain of mouse or one particular creature or another. But all of these things have been observed to some degree or another, and as Steve so eloquently pointed out in Boston last year, that very often there is a gender difference, so that males respond differently from females. So there's a great deal of complexity in this system. And this was really emphasized by the study of Liao, Ricky, Johnson, Diaz, and Nelson, published in 2009, which really rocked the calorie restriction community by pointing out that calorie restriction is by no means a universal phenomenon, that not even all recombinant inbred mouse strains exhibit beneficial effects of calorie restriction. So that, for example, roughly 1/3 of the population were negatively affected by calorie restriction. That is, they died earlier than those who had very minimal effects of calorie restriction, in contrast to those which we all have focused on, which exhibit an enhanced lifespan and increased longevity. So there is very definitely a background effect, an effect of genetic background here. And we shouldn't be surprised by this, because it's been known by the classical geneticists for a very long time. For example, the study by Leiter, Coleman, Hummel, published in 1981 in Diabetes, that the DB genotype has an effect on mortality, but the effect on mortality is entirely dependent on the genetic background in which that locus or which the gene is expressed. So that, for example, in C57 black 6 mice, there is no effect on mortality measured at 10 months of age of male animals. There's a small effect in female animals, whereas in the DBA2 background, the effect of the DB gene on mortality is very high. So there is clearly a very strong effect of genetic background on what we measure. Now, another very important thing which I've learned at Penn State, because I'm, thank goodness, being around some really good geneticists, it is that it is an accepted fact of life that genes hunt in packs. Now, this was really a revelation to me. I'd never thought about that, but it makes sense. These are from people who've studied genetics all their life and really know what they're talking about. Genes hunt in packs, and as such, the pack must be studied as a pack, and this by its nature poses difficulties. While it is always of interest to learn about the quirks of an isolated member, i.e. a single gene, the actions of these individuals as individuals is almost irrelevant for many purposes. They may have little bearing on the action of the pack as a whole. Now, this is quite at odds with the modern trend of looking for the gene responsible for this and the gene responsible for that. It is particularly pertinent in our study of aging if we're trying to find out those effects, those genes, those actions of the environment that affect longevity, a complex phenotype. So with this in mind, I and my colleagues, well, let's see. Yeah, well, just to emphasize the point, here we go from the DNA translation to the causal elements involved, hormones, tissue actions, and what have you, to a measurement of the phenotype. And the idea that since we now know there are epigenetic effects, the environment affects not just the expression of the causal elements, but feeds back on the DNA to affect transcription and translation. So we're dealing with a complex system, and from the aging perspective, it is a complex system whose interactions would be expected to change as the animal progresses over its lifespan. So the interaction of the genetic environment, of the genetic inheritance with the environment and with these causal elements will change as a function of the animal. And what happens when the animal is young would not be expected to be necessarily similar to the actions of these causal elements when the animal is older. So our general conclusions from all of these studies are that it seems likely that many different theories of aging contain elements which are pertinent to mechanisms of functional decline in some, but not all, gene-environment combinations. A complex system, we need to move away, I think, from recombinant inbreds and study heterogeneous populations and study them with different environmental combinations. And I think we need to get away from the idea that there's one universal product which results in the degenerative processes of aging. So the different theories will receive support from time to time as new studies emerge, and so may be considered immortal. And you've been very generous with your time in not peppering me with questions, and I was expecting a whole lot more, so I'll close at this stage and invite questions from you.